Pharmaceutical Processing, in conjunction with INTERPHEX, presents INTERPHEX Live.
Okay, last session of INTERPHEX 2019, and keeping with the theme, you know, we're still out there, we're talking about cell and gene therapy, and some of the, the components that go into that. And today we have our experts. We're going to be talking about isolation and containment in that application. And as you know, that has been touched on in several of the other conference titles, but I think this is one of the key pieces, especially in live cell therapies. Okay, we have with us today, we have Les Edwards, Vice President from SKAN, that's in Technology and Business Development. Marsha Steed, thank you for coming in from Bluebird. She's Global Microbiology and Contamination Control, so we've been looking for you for great advice. Marc, Director of Process Technology at CRB. And my old friend Christa Myers, not old, no emphasis on old, from CRB as well. Okay, so we're going to start off. And what I find is every time, at least it educates me, we like to ground the conference, or ground the beginning. And what do you mean by cell and gene therapy, Marsha? Why don't you lead it off?
Excellent. Good morning, everybody. Let's talk about what is cell and gene therapy. Make sure everybody's grounded here, right? So for cell therapy, we're really talking about transplanting cells, human cells, to go repair things that are damaged. So certainly there's a lot of T cells when we're talking about oncology and putting that cellular material into humans to fix a disease or something that's wrong. At Bluebird and many companies, we also do gene therapy, which does work side by side with cell therapy. So we're using a genetic material, your DNA, to go ahead and fix a disease that a patient has. So how do we do it? We all have genes, right? Whether we have green eyes or blue eyes, or we're tall or short, it's all about our DNA and our genes. So sometimes a part of a gene has a malfunction, something that's wrong with that part of the gene, and we need to fix it. So in gene therapy, we can fix that and take a healthy part of the gene and put that back in place and turn off that problem and cure the disease. So how do we get that good gene in where the bad gene or the mutation was? We need to put a carrier in place to carry that good gene in there. So we actually use, which sounds scary to some folks, we actually use virus. And we're able to manipulate that virus so that we take the bad disease-causing part of the virus out and make it a safe virus, which becomes a viral vector, where then we can take a good gene and put that in place and go cure the disease. So it's going right into the human cells. And this can happen inside the body or outside in the lab. So we could take some cells from a patient and go manipulate them in our manufacturing environment and then get them infused back into the patient, or we can infuse some of this straight into the patient and the cells will do their job in the body. Obviously, when we're making these, it's so important that we're doing this in an aseptic and sterile manner throughout, because we're talking about patient cells and everything has to be sterile so we don't contaminate something we put right into that patient.
And I think, Marsha, one of the other things that came up, and Mike and John did a real great job on it, I think people at times, when they're looking at it, they forget how involved and how protracted the virus vector process can be. I mean, we're talking a lot of time, a lot of effort, and this is the key piece of the starting material, as I understand it. Right? Did you want to comment on that, or?
Yeah, absolutely. And it takes so long to do the manufacturing process. A lot of times you might have open, exposed product that's out there for a long time, so it's so important. If you look at Annex 1 now, right? Annex 1 is all about contamination control and the revision, right, and how important it is that you're really having a good microbial control strategy. So I think even if it's something that you can do under a BSc in an open manual process, put it in an isolator wherever possible so that you're reducing that risk that you're going to have some contamination to it, because it is long processing times that it takes to do this.
All right, so so we get that, wanted to level up there, so, and we lead, so then that's the segue into the contamination. So contamination, I mean, so we're working on individualized material. I mean, this has got to bring contamination or cross-contamination to a whole new level. So what we're going to do is we're going to start with Christa. So what are the measures that we doing? How do you prevent all that kind of cross-contamination?
Well, see, so that's an interesting part of this, right? Because there's multiple levels of that. Yeah, you're trying to not, in the past, we've been talking about cross-contamination of, whether it's dirty into clean or and things like that, or whether it's drug 1 into drug 2. I don't necessarily want fentanyl in my Tylenol, nor Tylenol in my fentanyl, you know, so when we're doing those types of things. But this gets down to something even more close and more guarded. It's, I don't want Bob in my Steve or Steve in my Bob. This is patient-to-patient cross-contamination control. And so as we start looking at this, I have to be able to control not only the openness of that process, and if we can start closing parts and pieces of that process at that point, we can get to a safer functionality. Right now, we're using isolators as the part for that, but we also have to watch that facility as well. We've got viral vectors going on, and we've got to make sure that we've got contamination control not only at the isolator, but also in the facility as the operators are using that piece of equipment, in their gowning, in their procedures. We kind of act like it's magic, right? So, you wrap an isolator around it, we got it. There's still a whole lot of procedures that go with that, making sure that we're doing the decons the right way, making sure that we're cleaning the right way. We were having a conversation at dinner last night, and somebody had said that you know, the client called or the customer called and said, my isolator is no longer decontaminating anymore. What in the world? So they go and look at it. They're not cleaning their isolator on a batch by batch basis. They thought that the biodecontamination was magic. It's not magic. There's a lot of procedures around everything here.
That's a revelation. Oh, okay. So Marc, let's get back to the viral vector. I think let's talk about that in the context of what we're talking, cross-contamination. You know, there's a lot of things that can really go sideways on there, right? I mean, you know, we're talking about pre-infection, host. Why don't we get into some of that?
So
you got to turn it on. Is it on?
Is it on?
Yeah,
Yes, it is. Okay, thank you. Because of the sheer number of manipulations that take place, and the I'll say the number of patients that we'd also be looking at any given moment, the potential, the biggest risk in all of this, by the way, when you compare a cell and gene therapy operation to one of monoclonal antibody production. The beauty that we have in monoclonal antibody production is that we can sterile filter virtually every step of the way. Right. We don't have that opportunity here, that luxury here. So the need to truly isolate every unit operation from one another is critical, and the again, isolation technology is one of those ways. If we take a step backwards and we take a look at umbilical cord stem cell manipulations and operations, there had been developed, I know Thermo Fisher was involved in this, a fully closed operation in terms of they could do almost all the manipulations right there on the bench without the need of isolation technology. Sorry, Les, without the need of isolation technology, et cetera.
Okay.
And I think that is kind of where this industry is probably going to evolve over time, is the ability to have technology catch up to these operations, and hopefully truly isolate one operation from the other and truly isolate it from the environment.
I mean, the other thing is the operator. I mean, there's that risk as well, right? I mean, that's something that we forget, or
it doesn't matter if we're talking about monoclonal antibodies or cell and gene therapy.
Right.
Easily, the highest risk in any one of these operations is the human element.
Right.
And so we need to do whatever we can to isolate that human from the operations. And as you tour around the environments here, probably one of the most misunderstood or under-evaluated portions of all this is what I call transitions. So one of the advantages of isolators over, for instance, biosafety cabinet technology, is again in those transitions. As we go in and out of a biosafety cabinet, all elements within that environment, the human, any microbial contamination or particulate contamination in that suite, becomes a potential factor as you literally move your hand in and out of that biosafety cabinet. Obviously, in isolation technology, we've created the ability to create, in essence, an airlock that allows you to transition materials in and out of that isolator without issue.
Okay, so let's talk about the isolator a bit then. Let's ground it here. We kind of throw around biocabinet, isolator. Why don't we define terms, and right?
Absolutely. So there's one way of looking at this is in terms of generating different layers of boundaries in between your processes. So if you look at disposables and closed systems, and you even go back to aseptic manufacturing in general, a lot of what we want to do there is have sterile disposables that are single use, that are already closed processes anyhow. And then you have the isolator, which can act as a secondary containment and secondary safety level from a microbiological perspective. And then you have your facility and process flows to make sure everything is not causing or not giving a higher risk, and then your barrier system itself actually is going to assist you in that, right? Now, once we have a closed system, we're taking the humans out of the process from a microbiological perspective. We're providing not only protection to the product, but we're also providing protection to the operators as well. But getting all of these transitions, as Marc calls them, is really critical. How do you do that in a time-effective way? A lot of these biologics start off as a frozen specimen, okay? And you want to transfer a frozen specimen into a closed environment. Well, in order to get an effective decontamination process, does it need to be thawed first? Does your decontamination process cause any ingress or any problem with your cells? Are your cells inherently fragile? Or is some part of your process inherently fragile to something like a hydrogen peroxide gas or any other sterilant out there? So there's lots of questions to be answered, and each one of these engineered solutions that are helping you improve your process and improve your quality are also providing additional challenges that you need to look through in your risk analysis.
Can we just step through the primary, secondary containment aspect again? Just make sure I got that, right?
Okay, so your primary containment, again, would be your closed system, okay?
Right, okay.
Your product pathway, anything that's in touch with the cells or the end products, okay? Your secondary containment, a lot of people would consider that the isolator, but your isolator is actually your primary containment if you have any open operations, okay? In the ideal world, we will get to the point where we are able to keep this all in a closed environment. But just like right now, we're doing aseptic manufacturing in a closed isolator system in a lot of cases, we're still in a grade C environment, a little lower gowning than the typical conventional aseptic processing, but the same principles apply. And then again, the tertiary containment comes into how you design your facility, whether it's material flows and process flows, but also you may have some elements of containment in there as well. If you have a live virus that you might be using, that may need some facility protection as well, so you need certain airlocks and pressurization schemes in your facility.
So Christa, I mean, I'm hearing a lot of facility points here. So I mean, obviously you got to give a lot of thought to the design of the facility. It's not just let's drop the box in and be happy, you know?
Well, right. So it's a magic box, but it's not a magic box. There's still a lot of procedures that wrap around that. There are a lot of operational parameters that go along with that box. Reality is we've been doing aseptic processing forever, for a long, long time. And even in the biotech side that has been closed processing and large cell reactions for many, many years, there was still one open process, aseptic process that we were still doing. It was called sterile fill finish. It was getting that magic elixir into the last vial or syringe or whatever container we were putting it in. So the industry has a lot of experience with this. Now it's a little different when you start doing this and you start timing out how long some of these processes are open and the number of manipulations that have to take place. So not only do we have to watch the background and be able to plan the facility well, we have to actually go through some of the timing studies associated with how many steps is it going to take and how many isolators is it going to take and what is your throughput really going to need to look like? What does your cleaning protocol look like from patient to patient or room to room or time to time? All of those procedures add up. And we talk about microbial control. Right along with microbial control is particulate control. Every particle matters in these cases because this is the last step before it goes back to that patient. Everything we're talking about, it's about protecting the patient. My mom, my son, myself, your children, your family, those are the patients we're talking about. Let's not kid ourselves about this. This stuff matters. And so the rigorous procedures to be able to go through these processes to set us up for the facility working with the equipment, with the utilities, to provide a cost-controlled and good dose for that patient, that's what we're all driving towards. That's exactly what we're all driving towards. So we work very hard on these facility designs, the layouts, the people flow, the material flow, the waste flows. All of that plays in to how this comes together and whether that product is safe and effective for that patient and cost-effective to manufacture.
So I got a couple of things out. We'll step back and we'll do the flow on back up. I think the other part that I kind of stumble over here. Let's talk about the room classification, pressurization, let's get into that. Do you have where's the pressurization scheme, the differentials? You want to jump on that one, Marsha? You or Marc? Doesn't matter.
Sure.
Whatever you want to do.
I'll let Marc talk about it. I want to add on that point, and it ties into this point, how important it is that you're doing really good risk assessments now. Right. Not a check-of-the-box risk assessment to say I've done one because the regulators expect me to. Really go through every single step of the process and ask yourself what can go wrong and list out every single control you have in place. The cleanings, the sanitization, the pressurization, the classifications, everything. And then really score it in a realistic way that you ask yourself, do I need to put more controls in place to make sure? Everything is so manual at so many different places and there's so much human interaction. And then even when you get to the isolator, making sure that there's nothing you missed there on surface decontamination before AVHP. But I can't emphasize enough how important it is that we really take that design and then double check it with our cross-functional teams with a very well executed risk assessment to look at that.
So once upon a time, risk assessments were almost done at the end of things. Now, risk assessments are guiding us and leading us through these designs. It's not something you check the box and do at last. You start your process with that. You start your functionality with that.
The last ISP baseline guideline on biopharmaceutical manufacturing facilities really spells out the need to perform those risk assessments early. And I'll tell you, since the publication of that book, I've probably spent, I'd say 50 to 80% of my time performing risk assessments. And the beauty of it is when, especially in cell and gene therapy, most of the people coming into our facility, into our company to ask about facility design, et cetera, and what this facility may look like 2 or 3 years from now, or even in some cases now it's 7 or 8 months from now. Right. And they haven't really performed that risk assessment yet. And most of these people, not to put anybody down here, but most of the people coming into our organizations now are academics. So they've been doing this in the lab as, with animals, et cetera, and suddenly we're moving into the real world. And their initial idea or concept, because this is how they did it at the U of Penn, et cetera, is you'll see a facility with 4 or 5 biosafety cabinets in them. And they're literally manipulating 4 different patient pools at the same time in the same suite. And then they come to us and we perform that risk assessment and we very quickly show that the potential for cross-contamination from one process to the other or from one patient to the other, and again, never losing sight of environmental contamination and the potential of contamination from the operators themselves, we quickly start looking and diving into other design options, if you want. The hard part probably for less in all of this is people are used to working in a biosafety cabinet and you're looking at something that's $15,000 to $20,000. And they're looking at a facility that was probably in a lab space. Again, let's throw out a number, $500 to $600 an hour, $600 a square foot. And then SKAN walks in and they're talking about isolators in the $1 million range. And people right away will say, oh, no, I'm not jumping on that bandwagon. Right. I'm not ready for that. But with a risk assessment, and if you start really evaluating what the isolator does to reduce the potential risk to patient, and also how it removes the human element from it, Les made a comment here that we could put the isolator in a grade C environment. In Europe, you could put it in a grade D environment, which has just a major impact on how that facility is going to be run. Not to mention that now we can work in more of a ballroom approach because we can put multiple isolators in the same suite. In some of the earlier projects that I worked on, for instance, again, these people or these companies had multiple biosafety cabinets within that same suite, some of them face to face. and they were sharing some of the unit operations. So someone would literally take a vial out of that biosafety cabinet, put it in a centrifuge, which was in the common space, and then go back to that biosafety cabinet. You can imagine from a risk assessment standpoint, the potential of grabbing the wrong tube or someone simply with the wrong gloves, manipulating somebody else's flask.
See, my concern, let's talk about that for a minute. I mean, so, Les, I mean, there's the isolators, but in the context of what we just talked about, in a ballroom, in a whatever, common space, I mean, the pressurization schemes, the pressure differentials, how do you, as a supplier of these, you know, these are the bulletproof, you know, this is it, but I mean, it sounds to me like a misstep can compromise the whole thing, and I think it comes to pressurization. Can we talk about that a little bit?
Sure. I mean, pressurization is just one aspect of containment and cross-contamination. We have one client who is using a live virus to do their cell therapy process, and they are using, it's actually a gene therapy process, I apologize, but they want to have a certain level of priority for the aseptic piece of it, so they have their isolator running at positive pressure, okay? But their isolator empties not into the surrounding room, but it empties into a biosafety cabinet that's actually running negative, okay? So now that's another containment device. It's not an isolator, it's more in the restricted access barrier line of thought, but that's a containment scheme there, and every process is different. So you have to look at this. Again, everybody talks about risk assessments. A risk assessment is no good unless you understand your process. And that's why I'm actually very encouraged by a lot of the discussions and guidance out there, is that everybody seems to be looking at risk-based analyses. There's no right or wrong answer for a lot of these things. There are good and bad principles, but the whole world, I mean, talk about ballroom type of approaches. That was a bad practice before, okay? But now with a containment process, you can still establish, you know, versions of unidirectional flow in your facility, lines of lack of contamination or cross-contamination, so you can have a train with a logical process flow that doesn't cause these problems. And with, you know, pressurization schemes with the rooms, pressurization schemes with the isolator systems, they are one factor in the safety aspects of this for sure, but that's a very small piece of the pie.
Right. So, Mark, what I want to get back, so let's get back to flow. I mean, you would say, take it from here, put it over. I want to get a sense of, against the backdrop of the pressurization. Let's talk about flow. Let's talk about personnel, material out, waste material. I mean, that's a, you know, can we just?
It's a key element. You're absolutely right.
Yeah. So.
No, absolutely. I mean, the key to all this is, I mean, there's a statement in the CFR and virtually all the guidelines, and it's the statement of you must do what you can to prevent mix-ups. And again, having and please don't misinterpret what unidirectional flow means. It doesn't mean that you have to go north-south all the time or east-west all the time. But no, seriously, you laugh, but if you look at some of the academics that are presenting to us and saying, hey, I want to get into commercial manufacturing, they're thinking east-west, you know, north-south. And what we're trying to say is, as you take your initial process flask, let's use simple terms, your initial process flask, and you're working to the end result, to the fill finish type operation, you want to make sure that all of those steps in between, in a sense, are virgin every time. It's not necessarily the manipulations of the operations that are the highest risk. It's how did you get the environment prepared for that unit operation? And so, getting back to the risk assessments, probably the most important discussions that you must have in performing the risk assessment and in terms of determining the correct flow of your process is to look at, how did I sanitize this thing? How did I clear this thing? How did I set up this thing? Et cetera, and making sure that all those unit operations are performed in a very systematic fashion. And again, making sure that if there is some, I'll say, common equipment used, what is the transition between Bob's product and Fred's product? And how did you return that system to a virgin state prior to making that manipulation?
So the process is not only just the process in that state, right? So you have to watch cleaning, you have to deal with waste removal, gowning becomes important, glove change becomes important. You know, you start trying to limit those access points or limit those contamination points, and it drives back to facility design over and over and over again. You've got to be able to work with a team that can really have the experience to walk your team through that and be able to come up with what's the right approach for my process, because they're not all the same.
Absolutely. And then when you tie into that, right, that material and personnel and waste flow, and don't forget about what flow are my environmental monitoring folks who go in and monitor, are they going to break the unidirectional flow, or do they have to be able to get to the samples? What about my people who come in and clean the clean rooms probably at night, right? Are they going to go unidirectional or break the flow? And what's okay, what's not okay? But in this manufacturing and cell and gene therapy world, we use so many sterile, single-use disposable items. Those items need to get from our warehouse into the manufacturing rooms, through the corridors, and oftentimes into our BSCs or into our isolators, which will get more decontamination. And that is one of the biggest risks, and when you do these risk assessments, it comes out, or the EM that you see in some of these spaces. So you can have wonderful flows, but if you're not doing the right sanitization, getting these materials from a cardboard box in a warehouse in, you're gonna end up having some molds and bacillus in your clean room. So it all ties together, and the risk assessment should really help highlight all of this.
So Marsha, if we had a list, I mean, as a process owner, okay? If you were going to list the biggest challenges when it comes down to this, I mean, how would you start to take that down? Why don't we, I think if we step through that, that'll be a really good piece of information.
For definitely, you know, some of those challenges are space, right? It all goes back to a good design. A good contamination control strategy starts with a very, very good design and understanding what space that you're going to need so you're not overcrowding, right? That you get your right flows down properly so you're not trying to make something work in a space that maybe it wasn't designed for, it doesn't work very well. Then putting the right materials and containment into place. That also goes through, because certainly I'm a microbiologist, so I see a lot of the challenges on where do we end up getting contamination, and so much of that has to do with material transfer. 90% of the time, right? We have two things. It's the people, because it's so manual, and sometimes it's so open, so we're going to get skin organisms. It's going to happen no matter how much gowning we do. But we also see a lot of hits from not using the right kind of sporicidals or the right cleaning agents as we're trying to get these materials through many different locations, and they sit in different places for a long period of time. Do they need to be resanitized before they're used because they came into the room 4 weeks ago?
So Les, I mean, one of the things that we're talking about as that, I mean, you kind of touched on it when it's thawed, unthawed. I mean, I think can we talk about that a bit more? Because it's come up on several of the discussions we've had, maybe.
Right. So any sort of decontamination process that you use is got to start off with a proper understanding of what your challenge is to it regarding, you know, is this an item that has had some bioburden control because we've kept it in the grade C environment, we've had a low bioburden around it, we've removed an outer overwrap or something like that, and we're just decontaminating the outside of a pretty low bioburden item. If you need to do a fully validated sterile transfer into your critical aseptic zone, you know, obviously we use hydrogen peroxide gas as a sterilant for most of those environments, a very well-studied, well-known sterilant out there. But the hold times and everything that you're in there that are required for that are automatically respected when you have a piece of equipment transferring your item into an isolator. Okay, so the wipe downs of items where, you know, people are using just sanitizing agents like alcohol and things like that in a lot of lab environments, or they're using sporicidal items and they think, well, that's great, I'm using a sporicide here, therefore I'm going to be good transferring it into an isolator. But if you look at the bottle on the outside of the sporicide that says, you know, 8 to 11 minute contact time, and they need to get it in there in 2 minutes, and they think that if you wipe it twice or wipe it faster somehow, that it'll kill the organisms more, or use a hammer to.
Or use more of it.
Yeah, more of it. More is
of it.
better, right?
Douse it, right?
It's not. So you got to have good science around that, and the more automation and more sophisticated transfers that you have, the better job you're going to do on doing all these critical transfers. And when we talk about things like a cold chain of events, you have to look at that as well. If you take a very cold object and you put it right here on this table, okay, it's going to have a lot of condensate that starts to form on that surface. Okay, with our fogging processes that we use, we're depositing hydrogen peroxide directly onto those surfaces, so that helps, but that's also being blocked a little bit by the condensate that may be formed there. Okay, so you want to start off with a clean, dry item. Okay, and if you can't start off with a clean, dry item, you have to do some process development around bioburden control and/or bioburden study to know what your risks are. And if you know you're not going to do a perfect job and you don't do a perfect job, that's not necessarily wrong, right? Because some people say, well, I can't validate that more complex process because I have this technical challenge and I can't get perfect results. So I'm going to use this really bad process of just wiping it down and not doing any validation at all. And so they're almost ignoring the problem. And that's comes down to, again, we're getting back to this risk assessment.
Right.
You got to be smart about this stuff and realize that you want to do the best that you can using the best available technologies, and that always gets you to a better place.
So one of the things that always surprises me about that are the little details. And there was a 43 that came out a couple years ago. It's not uncommon for us to put a bottle of WFI into an isolator to be able to do wipe downs during process and things like that. The 43 said that the manufacturer did not have control of their biodecontamination cycle because when they put that bottle of WFI in, they did not remove the occluded surface. ... It goes into a little bit more. They had not taken the cap off the WFI bottle. So the WFI bottle has a aluminum seal on the top of it. Had not even thought about it until that point. Yeah, they're absolutely right. That was an occluded surface that was not getting biodecontaminated. It's tiny little details that matter when you start using these technologies. They're out there, they're stable, there's a lot of knowledge in the industry about them, and even the transfers in and out of these pieces of equipment, we know the details. We know what can be used for those types of things, but it still is not an excuse for bad procedure or bad aseptic technique. You still have to know what you're doing and to be able to make sure, again, that you can protect that patient with what you're dealing with.
Getting back to your cold chain question, because it's something that I've been thinking about for quite a while, and in my opinion, so if you take a look at a cell therapy, especially autologous cell therapy, you're looking at, people will say it's anywhere from a 20 to 30 day process, and 10 of those days is cold chain.
Yeah.
Eliminate it. I think in the future that's where we're going to end up, is we're going to bring the patient to the process, or the process to the patient. And so having centralized facilities, which is, I think, one of the highest risks actually in this whole operation, is again to mobilize the manipulations and operations. And right now we're talking some very small scale operations, which lend themselves very well to a very small environment. Again, with extra protection, making sure that you got the right tools in place. But I literally see a future, and not too far from now, where we're putting isolators within hospitals and with a McDonald's type setting for the patient and their families, and we're essentially doing all of the cell therapy right there in the hospital.
I agree. And right, in order to get these wonderful, wonderful products to patients all over the world, we have to get to that kind of technology, right? How do we get it to far places in Africa, or who knows where around the world, to remote places that they don't have the luxury of just coming into a hospital in New York City to get their treatment? So we have to, with that commitment to patients.
The other thing, Mark, I wanted to, on that, I didn't have a chance to say, but to me, and as a process owner too, Marsha, it's, we're going to go about when it's live cell. Operative word is live, and things we do kill cells because we want to, right? But there's the good guys and the bad guys. So how do you not obliterate the good guys? You know, I mean, what levels? I mean, it sounds to me like we're in an area now, and I'm sure it's been discussed, but for our audience, I think, you know, hydrogen peroxide, that's really basically a pretty bad thing. I mean, so how do we balance that decon against what we're doing, and how do you make sure it's there?
Well, obviously you can't expose the cells to any
Right.
VHP type process. And again, I mean, cells don't like DMSO. They don't like the cryo process. And so we got to do whatever we can. And again, so if we can eliminate some of those steps, and again, time is one of the, cells just don't like being outside my body.
Right.
And so, you know, the more I can do, the closer I can get to extraction and re-injection of that engineered cell, the better. And again, so this is where we're going in terms of bringing that technology right there to the hospital and to the patient. And again, I think that future is not that far away. And there's some of the facilities, for instance, that we're designing right now that get us there pretty quickly. And we talk about the cost of these treatments, by the way. We're looking at anywhere from I'm being generous when I say $500,000 to over $1 million, and sometimes multi-million. Right. And a lot of that is all of these manipulations that we're doing just for cell preservation.
But for a cure.
Absolutely.
In many cases, it's for a cure. And that's the incredible part of this.
But where I'm going with this is I can see these technologies, if we look at it from a holistic standpoint and we start talking about bringing these operations back to the hospitals, I see us getting down into the less than six-figure costs very quickly.
Right. See, I didn't want to get into the pharmacoeconomics on this because you always hear people talking about 10 years of therapy versus a one-shot therapy. Yeah. The numbers don't really, they don't help us either way. I mean, right now it's not that doesn't drive decisions.
But when your child is suffering,
agreed, but
it's hard.
You're going to be in for the $500,000 either way. I mean, okay, and I don't think there's a person that walks that doesn't say, it's my kid, I'll do whatever it takes. My question is this, though. Let's get back to, and I think more, are we talking about, if you take this thing full circle, the way I think of it is, it's like the guy or the woman is coming in for a dialysis treatment. I mean, ideally, that's the deal, ain't it? I mean, do you see it getting there? I mean,
absolutely. I mean, again, I get back to the umbilical cord blood. And actually, if anyone ever gets a chance, it's literally down the street in Long Island here at the New York Blood Center. It's the first fully licensed umbilical cord blood center on Earth, actually. And that operation is fully closed, except for one step, is the DMSO addition. And it's because they actually manipulate their operation using a multi-dose DMSO application or vial, which they must perform in a biosafety cabinet, but they're doing it in a biosafety cabinet, which literally, when I did the risk assessment on that project, represented the largest risk to this operation. In their case, using a single dose DMSO, which would be per patient, they literally have a fully closed process, which we could literally take care of 25 patients right here in this little space, sitting on a bench. No, sorry, Les. Yeah. No isolators.
Right. So I think for us to get there, we're going to, each of us in the industry, both as researchers, engineers, equipment manufacturers, we're going to all have to push for those closed processes, push for the better than what I'm seeing processes in these events so that we can get there. But it means that all of us as engineers, as designers, as equipment designers, we've got to start thinking about that. How can we take some of these med device products and cross them into our industry to make sure that we can get down to some of these small functionalities?
And if I can just jump in, and along the same lines, respectfully fight my buddy here about, isolators are important. No. It actually, as a concept, though, it's a very attractive approach, because if you think of, the ultimate distributed manufacturing facility, wouldn't you love that to be a controlled, little protected facility that's well designed? And so an isolator isn't going to be a box around your process. An isolator is going to house your process. It is going to be your process. And that's a major, major part of it. You talked about, these decon processes, and the cells don't like hydrogen peroxide. That's why we've had to develop methods for not only getting kill, but the residual removal is huge. And we've had to develop a lot of science to get down to the parts per billion range of hydrogen peroxide so it doesn't interfere with cell growth and tissue growth.
So
And-
Marsha,
yeah.
excuse me, one of the things that was said in one of the earlier sessions, in fact, was, okay, we go through all of this, but once they're in, and it's going to get back to process qualification or
That's right.
process once we administer, it's vein to vein, so on the tail end, it doesn't stop there. I mean, this thing continues. So
That's right.
post-production, I mean, can you just shed some light on that stuff?
Sure, on cleaning up your isolators and
Yeah.
making sure, right, you haven't left anything behind. So a lot of this manufacturing takes place at contract manufacturing organizations, right, at CMOs. So if I'm going to put a bluebird product through a CMO, I want to make sure that I'm not picking up whatever X company went through that isolator or manufacturing process before. It goes on, and you have to make sure that you have covered on all ends, right, that you're able to. And the science is fantastic, so that there's great assurance that, yes, I'm not going to be carrying over some virus from that last company, and that's going to go through. Or if I'm a multi-product facility, that I'm going to take one product and carry it over to the other. It goes on and on.
Well, and even for your single-use components, you know, the quality groups are doing massive audits of these single-use components that are coming in to make sure that they don't bring in extra bioburden, to make sure they
That's
don't
right.
bring in extra particulates.
That's right.
So that's still a part of the responsibility of the company that has their label on that vial. You have to audit your sub-suppliers, and you have to have control of that path.
But what about release testing, Marsha? I mean, you know, we're used to an environment where we know what it is, we put a date on it, we track it, we know from so how do you, in this environment, that's where I'm getting to,
Yes.
is it's vein to vein. So does that come to play, or
It does. And so, of course, I'm the microbiologist, so I care about sterility, endotoxin, right? Safety tests that we would normally do for any normal parenteral or product, but it's important. And sometimes, if you're getting a cell therapy and I'm taking in patient cells that are coming into me through an apheresis and then it's going to go back, could be contaminated coming through the door, and now I've just entered a contaminant in. So, you know, a lot of questions, and a lot of regulators want you to do that sterility test on an incoming patient sample, so you know. It might be just to help you for an investigation, so that at the end, before you put it back out there to the patient, but I can't wait 14 days. I need to get this back vein to vein right back into that patient and cure their disease. So I need to do rapid sterility testing, right, which is very much so accepted by the regulators and expected that you'll do that. I might find out I had a positive, and then sometimes then you have to say, here's what the ID was, and the doctors are going to treat with an antibiotic. But it's a very complex system that we have to do, and we keep trying to get better. Rapid methods are helping us get these answers sooner, but you have to make sure from a patient's safety perspective, and then of course there's many other tests that fall outside of my micro, but you know, potency and all kinds of cellular tests that we have to do to make sure it's safe. And you get into complex issues of, we don't have that much. So how am I going to test the quantities if I go to my normal compendia that says what I would normally test, because they were written for thousands of vials that are made, and you have to come up with good risk justifications, and we're getting there together as an industry, but it's a complex process with lots of testing.
It's complex. I mean, bottom line is, right, the input could be all over a lot. The output subsequently is going to be all over. So how do you know if that's what you want, Marc? I mean, you did the risk assessment. So when you test it, I think it's okay? I mean, you know, you follow what my
No, I don't understand
Right,
the
in other
question.
words, how do you put a specification on something that could vary that much? Is that easier? It isn't, is it?
I'll say, yeah.
I'm going to defer
you
to
know what?
Marsha, this one.
This is very, very difficult, and sometimes, right, you might come up to, wow, I set my spec range too tight because I only had a little bit of clinical data, and now what, right? It's a horrible thing to say, hey, I really did pass, or I have to reject this, and a patient can't get this life-saving treatment because I failed, when really maybe I shouldn't have failed, right? It is very complex, and it is something that the industry is struggling with, to get the right specs in place, to get the right testing in place, so that you balance out making sure we're not going to hurt a patient by putting something wrong that doesn't meet something, and using limited data that you have to truly set up your right quality control specifications in place. It is something that the industry and every company I know struggles with right now to get it right.
I'd like to expand on the word control, because one of the advantages, and this is, you know, Les really hasn't talked about this very much, but I think it's one of the biggest factors in terms of the beauty of isolation technology, is what is my working space? And so when we talk about control, you're talking the control of the process, control of the product, control of the environment in which you're working. And the problem is, I'm sure if there's anybody here that's been in the monoclonal antibody world, is when you've got that product that you know on the table, there's absolutely nothing wrong with it. Right. Oops, my grade B environment had a deviation in it. And you're in deviation hell for the next 30 days before you can release product, not because the product has any issue with it, but because your environment had an issue with it. One of the beauties of isolation technology is you've got a very small space that you need to control. And the ability to control the grade A environment within an isolator is, I won't say a slam dunk, because it never is, but it's almost a slam dunk compared to trying to control the environment in which you're working with, in which there are 2 or 3 human beings walking in and out. You know, Martha walks in one day, or Marc walks in one day, and that environment changes totally, just for the fact that the 2 of us aren't the same people. But again, within that isolator, when you remove that human element, control of that environment is so much easier.
Well, it's one of the interesting things about that type of thing, right? So on the aseptic fill finish side of things, we're used to doing these media fills and media fills and media fills to prove what we're doing. And the space where we're doing this type of processing doesn't necessarily understand that or have that to be able to verify that, you know, Marc got a sunburn yesterday and he's shedding extra cells. He shouldn't be in here. That's the kind of control that our industry expects, and the isolator does become the magic box in some of those cases, right? In that it becomes that tighter controlled environment. There's a reason we call it an isolator. It isolates some of those humans from that process. Now, we still have to follow our right gowning, we still have to follow our right procedures, we still have to follow our right cleaning, but control goes all the way down to cleaning the room and of the isolator. It goes all the way down to training those operators. One of the biggest things we're going to be dealing with is training these operators. Good aseptic technique, good gowning, good procedures, good thought processes. It's all going to be important.
Okay.